All right, everyone, let's get started. Good afternoon, everybody. Welcome to the North Carolina Museum of Natural Sciences. Thanks for coming out here today and visiting us here at the museum. Or for those of you I recognize on the first floor, thanks for coming to work and being with us here today in the museum. Every Wednesday at noon, we host the Lunchtime Discovery Series. It's a partnership between us here at the museum, our digital media team that broadcasts the program out onto the internet, and us here in the Daily Planet Theater, and North Carolina's Department of Environmental Quality. The Office of Environmental Education works to source and bring in and invite our fabulous guest speakers into the globe here inside the museum, where we get the chance to learn something new, right? That's always fun. And what's great about this series every week is that we're bringing in, most of the time, local experts who can share with us what's happening in North Carolina, what scientists and other professionals are thinking about these issues, what they're passionate about. And we always have a great discussion afterwards, after the presentation, with audience Q&A. So today, I expect will be no different at all. From the looks of the topic, though, it might be a little bit scary. We've got the words toxic, but even scarier, cyanobacteria. Oh my gosh. You've heard about these things in the news, right? Yeah, anytime this stuff pops up in the news, it's always headlines. Could be thought of as scary stuff. And fortunately for us, and I hope you'll agree too, there are experts like Haley Plaas at UNC Chapel Hill who are thinking about and working on these issues so that they don't have to be so scary to us. Please put your hands together and welcome to the stage, Haley Plaas.
Hi. Thank you all so much for having me here today. I'm really excited to talk about this scary topic, cyanobacteria. So without further ado, just to give you a quick overview of my talk, first I'll be talking about factors that promote cyanobacterial blooms in North Carolina as well as across the world. And I'll also be talking about factors that promote toxin production and different toxins that are produced by cyanobacteria. And then I will talk about the potential aerosolization of these toxins and cyanobacteria cells in general. So we're going to be looking at that this upcoming summer. And then I will also talk about just public health in general and why this is a threat to our ecosystems as well as our human populations here in North Carolina. And then I'll give the specifics for the upcoming research that I will be conducting under a joint fellowship with APNEP and NCC grant next summer in the Chowan River and Albemarle Sound. So without further ado, just to give a little background information about me, my interest in aquatic sciences actually began when I was very, very young. So that's a picture of me that you can see right there. I grew up in the Midwest. I was born and raised in Kansas City, Kansas, but my parents were living in Ohio, in Cleveland, when I was 3 years old. And so on the weekends, my mom would take me to SeaWorld to entertain me. And as you can see, I was glued to the dolphin exhibit. And what started as a fascination and obsession with whales, which is actually still a fascination and obsession with whales, turned into a passion for studying how algae influences public health. So I graduated from the University of Miami in May with a degree in biology and marine science, and now I'm starting my PhD at UNC in the Environmental Science and Engineering program in the School of Public Health. But my interest in algae and how algal toxins influence public health first began when I was interning at the Mote Marine Laboratory in Sarasota, Florida. So if you've heard of the Mote Marine Laboratory, it's probably because you've heard of the red tide blooms that have been ongoing in West Florida, on the west coast of Florida, for the past couple summers. And so during this time that I was there, it was the summer of 2018, South Florida was actually experiencing a dual toxic algal bloom. So we had a freshwater cyanobacterial bloom in Lake Okeechobee, which is a giant freshwater lake in central Florida that's at the head of the Everglades. And we also had the ongoing red tide bloom, which is a marine dinoflagellate that is endemic to the Gulf of Mexico on the west coast of Florida. So the people living there at the time were really concerned because this red tide bloom on the beach was causing beach closes. We were getting a lot of dead fish washing up. The brevetoxin that's produced by the red tide is a potent neurotoxin, and it's very irritating to people when they breathe it in. And so what happens with this brevetoxin is that it's easily aerosolized. And when I say aerosolized, I mean it's translated from the water to the air via breaking waves. So when waves break, they entrap air bubbles beneath the surface. And then as those air bubbles burst at the surface, this can cause chemical matter and biological matter that's resting in the water to be translated to the air. And so that process is called aerosolization, and it generates aerosols that contain a liquid or a solid floating in the gas. But getting back to what we don't know is, we know a lot about this with red tide, and that's where my interest in aerosolized toxins first started, but we don't know as much about it in cyanobacteria and with cyanobacterial toxins. So, getting to the culprit of these toxins, cyanobacteria. So there's several factors that can promote cyanobacterial blooms, but the phytoplankton itself is actually a healthy component of the basis of a food chain. So cyanobacteria it was actually the first organism on Earth. So it's very old, it's very hardy. There's many different strains of cyanobacteria that can grow in freshwater, in estuaries, and also in the ocean. But the cyanobacteria species that we focus on are actually freshwater. So those are ones that we want to be the most concerned about in North Carolina. And so, is there a way that we can, sorry, I just feel like keeps going in and out. Awesome, is that better? Okay, great. So there's a couple different factors, as I said, that can promote the blooms of these cyanobacteria. So although they are normal and healthy in the basis of our food chain, they're just an algae. Under the right conditions, they can condense into these dense surface cones that you would think of when you think of an algal bloom. And so my lab, the Pro Lab, which is run by Dr. Hans Perl, for nearly 40 years now at UNC Chapel Hill's Institute of Marine Science, we study the influence of humans on cyanobacterial bloom growth and dynamics, and we also look at how these blooms influence human health as well. So we're looking at how we affect blooms and in turn how blooms affect us, since human health is so delicately intertwined with ecosystem and environmental health. So first, how do humans influence cyanobacteria blooms? And the first way is through climate change. So as our waters are heating up, we're getting increased temperatures in the atmosphere that's leading to increased surface water temperatures in our lakes and oceans. This leads to stratification in the water column. So when we are seeing these dense blooms, they're not as easily dispersed because we're not seeing mixing in a water column. And also this heat may favor cyanobacteria as opposed to other types of phytoplankton. So for this reason, these cyanobacteria blooms could be increasing in their frequency and duration because of increased heat. And then the second way that we influence cyanobacterial bloom growth is through nutrient loading or eutrophication. So nutrients can be delivered to these waters from rainfall or runoff. So a lot of this fertilizers that we use on agriculture, on farms around North Carolina, and also in our backyards can fuel the growth of these blooms. So since they are tiny plants, they can grow as any other plants do. And so that fertilizer that we're using to treat our lawns, our crops, could be used to allow these cyanobacteria to condense into the blooms. So how do we study this in the Pearl Lab? And the first way is through routine monitoring. So we have ongoing projects in the Neuse River and Pamlico Sound. And we just want to monitor the phytoplankton communities and the cyanobacteria that are living there throughout the year, see what they're doing, see what they're eating. And then we also have ongoing study at Lake Erie and also at Lake Taihu in China. So those are some places that are experiencing recurrent cyanobacteria blooms that we kind of want more information about. And the other way that we do it is we collect water during bloom conditions, and we have a series of bioassay experiments. And what a bioassay is we basically divvy all of these water samples up into cubitainers that look like big milk jugs, and we treat them with different amounts of nutrients or different dilution factors. And so what we're trying to see is how much does this bloom need to be diluted in order for it to stop growing so densely, or what nutrients are these cyanobacteria thriving on? What ratios of certain nutrients of nitrogen and phosphorus and carbon, et cetera, are these cyanobacteria using to bloom so densely? And so what we hope to do with the information from these studies is to work backwards from that in order to inform policy and mitigation strategies. So if we can figure out what's making these cyanobacteria tick, then we can ultimately decide what nutrients we need to target to reduce the amount of runoff that's reaching these cyanobacteria and allowing them to grow so densely. Okay, so where is this occurring in North Carolina currently? And cyanobacteria blooms are really growing all over the state. So this is a map that has been modified from the North Carolina Department of Environmental Quality, which it's like, who's from there today? Show of hands, lots of you, awesome. So I'm sure you're familiar with this map. So there is an interactive map on their website. I have a link at the end that you can access this map, but all of the dots in red are cyanobacteria blooms that have occurred since 2012. And it's important to note that these blooms occur in the early spring to late fall because cyanobacteria require enough temperature, sunlight, and then of course rainfall to deliver those nutrients to grow into these dense conditions. And this is from routine monitoring by the water department of the NCDEQ. But this isn't all the cyanobacteria blooms that have occurred in North Carolina. There's of course going to be blooms that we don't hear about that might be occurring in people's backyards, in retention ponds, and little creeks around neighborhoods. And then this is where it's really a concern for people's safety, and livestock safety, pet safety. So although this does seem like a large amount of cyanobacteria blooms, these are the really big water bodies that are constantly monitored. It could be occurring in other places as well. So getting to the meat of why this is a public health concern and ecosystem concern, which I really think really fall into the same category. If it's affecting our ecosystems, it's going to be affecting humans in some way. The first reason is through a loss of aquatic biodiversity. So these blooms are dominated by 1 to 2 species of cyanobacteria. And so this can reduce the other amount of algae and phytoplankton that you would normally see in these aquatic ecosystems. And so this isn't good for the rest of the food chain, and it can also lead to fish kills and harm other larger wildlife. So you don't want to see that because this leads to a decrease in the integrity of the ecosystem, which is never a good thing. Also, it decreases water quality or water clarity greatly. So you see a great increase in the turbidity of these waters when you have these dense cyanobacteria blooms. So this can be bad for predator-prey interactions, fish in these ecosystems, and it also doesn't allow other macroflora such as seagrass or salt marshes to receive the amount of sunlight that they need when you have these dense algae growing on the surface. The drinking water issue becomes a huge problem in developing nations where people are using water from their backyard as their main source of drinking water. So you don't want to see rampant algal bloom growth in places where people rely on the water in their backyard as their drinking water. And same goes for recreational activity. These blooms can affect the use of main fisheries and also recreational places. So no one wants to be around a really dense algal bloom. No one wants to fish there. It often stinks because as these dense mats of algae die off, bacteria will come in to break them down. And so that creates this really bad stink and also pulls a lot of the oxygen out of the water, leading to anoxic conditions, which can also kill fish. So overall, algal growth in general, when it runs out of control like this, can be really bad for an ecosystem, but that doesn't even include algae that are producing toxins. So any runaway algal bloom can cause these problems in an ecosystem, but harmful algal blooms, and specifically cyanobacterial harmful algal blooms, which I will refer to as cyanohabs, are characterized by the production of these toxic secondary metabolites, which are the focus of my study in the public health school. So there's a whole variety of cyanobacterial strains that produce cyanobacterial toxins or cyanotoxins, and they can impact people via ingestion. So if people drink water that has these toxins in it, it can attack people that way. Also, if you have really high concentrations of cyanobacterial toxins, you can even see dermal uptake. So if you have really concentrated toxins, you don't want to be touching the water. And then also it can accumulate in fish tissues. So various cyanotoxins have been shown to accumulate up a food chain And then when people eat contaminated fish, you can have some really bad side effects that way. So we know a lot about how the ingestion of cyanobacterial toxins affects people, but what we don't know a lot about is how the inhalation of cyanobacterial toxins affects people. And before I get to the topic of the inhalation of cyanobacterial cells and toxins, just to talk about a little bit of the factors that we have hypothesized that might lead to toxin production in cyanobacteria. It's actually a little bit confusing. We have a hard time kind of piecing apart what environmental factors may lead to toxin production in these various strains of cyanobacteria. It can vary between time, it can vary between areas and regions, and so there have been a couple different theories that have been proposed that might influence the production of these toxins in cyanobacteria, some being interspecies competition. So it's theorized that cyanobacteria may produce toxins to ward off predators or grazers. And then it also has been shown that when you sample the cyanobacteria that's producing a toxin, many times it's demonstrated that when you start culturing it in a lab, it stops producing toxins. So there might be some stressor in the environment that's causing these cyanobacteria to produce the toxins, which is why they're doing it. And so that's something that I'll be looking at as well in Eastern North Carolina for my studies. So one toxin in particular that has been demonstrated in the Chowan River, which is what I'll talk about in a minute for my upcoming research, is microcystin. And microcystin is produced by a whole suite of cyanobacterial genera, but including microcystis, which is one of the types of cyanobacteria that we study commonly. And microcystin is a liver toxin, so it primarily acts in people's liver. And you can see those two little red circles there indicate the variable side chains. So this chemical, microcystin, can be produced with different amino acids in those two positions. And that's of large interest to me because I think that these different positions could lead to differences in transport through the environment. And if we can better understand how these toxins are moving through the environment, then we can better understand exposure pathways. So put that in your back pocket because finally, getting to the inhalation of these cyanobacterial toxins. So as I mentioned, the main mechanism that is through bubble bursting. So this diagram here just kind of demonstrates what's going on as those waves break, which we can see in large lake systems. They entrap bubbles beneath the surface, and when those bubbles burst, they can release these algal toxins into the air. And so what we really don't understand and dissolved in the water. So that's another question that I'm hoping to answer. But it's important to know that the inhalation of algal matter could cause problems even if it does not have toxins. So we define aerosols as PM10 or PM2.5, and PM stands for and below, and PM2.5 is any particle that is 2.5 microns and below. And the smaller the particle is, the more adverse the health impact because it can settle deeper into your lungs. And so if it settles deeper in your lungs, then it can cause problems with your alveoli where you're having your blood oxygen exchange, any sort of aerosolized matter there. Predisposition to respiratory illness later in life. So we would like to know if these really dense cyanobacteria blooms that are resting on the surface are becoming aerosolized in one way or another, even if there are not toxins presented in these aerosols. And so, let's see here. The other interesting thing about this is that not even from an inhalation standpoint, the aerosolization of algae could be of interest to us because it's been demonstrated that this biological matter can be transported over really long distances. So these algae might actually be serving as cloud condensation nuclei in the atmosphere. So this could have even farther climatic and weather impacts. And since, these cyanobacteria blooms are sitting on the surface, I find it very likely that when we're seeing wave action during these bloom events, there is some sort of cellular matter that is becoming aerosolized if the toxins themselves are not becoming aerosolized. And of course, if the toxin is maintained in the cell, then you would assume that the toxin could be inhaled as well. So this is something that we want to generate data on. All right, so what we currently know about the aerosolization of microcystins specifically is there's a couple studies that have been conducted. So there's a lab group out of the the Florida Gulf Coast University that is very close to the Marine Lab actually, who started looking at this problem when I was living and working in Sarasota at the time. And they have demonstrated that microcystin can be detected in aerosols up to 2 miles from the source. And this is consistent with findings with the red tide toxin brevetoxin. So they have not yet to publish any data on this, but they did report that last summer. And then as well, there have been some studies conducted to look at the inhalation of microcystin in mice. And what was interesting is that they looked at a very acute dosage of this inhalation of the aerosol, but I think what we need to be concerned about right now would actually be a more chronic exposure to it. So they were exposing these mice to a very large concentration of microcystin, and they weren't actually seeing any activation in the liver of these mice. So there was no, this toxin did not cross the blood barrier when it was inhaled. Rather, it was leading to nasal lesions in the mice. And so although these nasal lesions were response for an acute exposure to this toxin, I think that our concern, if we need to be concerned, would be actually a chronic exposure to people who are living near these areas experiencing these blooms. So getting to the specifics of my research on the Chowan River next summer. So you can see here the Chowan River is circled in red. We have a large concentration of cyanobacteria blooms occurring over the last 5 years in this area. And this area is a really interesting region to study for a couple reasons. The first being its history. So in the late 70s and early 80s, this region was experiencing toxic cyanobacterial blooms, and thanks to research done in part by my advisor, Dr. Pearl, they were able to successfully mitigate these blooms. So they found agricultural runoff to be the culprit, they cut some of the nutrients out, and then we didn't see these toxic blooms for a handful of years. But for some reason, in the past 5 years, they've made a resurgence. And the other reason that this region is really interesting to study is because of the salinity gradient. So the Albemarle Sound does not see much exchange with the Atlantic Ocean due to the Outer Banks system. So this allows the cyanobacteria blooms that are originating in the Chowan River to persist farther down into the Albemarle Sound. So that's really interesting for us because we can kind of study how the salt gradient affects toxin production in the cyanobacteria, and it's also interesting because it can affect more people because of this, so it can spread over much farther distances. So, as you can see that I have written on the slide, the microcystin concentration in this area was actually measured up to 300 micrograms per liter last summer. And this is 300 times the recommended daily intake for drinking water. This is super toxic water we're talking about. The blooms are really active here in the last summer, and we definitely want to find some solutions to this problem because this is a key fisheries and recreational use region. So this is not looking good for the people living in the area and also for the people visiting this area for ecotourism. So the aims of my study next summer will firstly be to quantify any amount of cyanobacterial toxin that might be in the air. And we're going to focus specifically on microcystin since this was the toxin that was being demonstrated in this region last year. So we're gonna set out some pumps, which I will show in a minute, and we're gonna see if we are detecting any amount of toxin and aerosolized cellular matter in the air. Secondly, we're going to look at the role of salinity on toxin production on these cyanobacteria, and we're also going to look to see if The salt changes the phytoplankton community composition, meaning what kind of algae are there in response to the increasing salt gradient. And we're also going to see if there's any association between the type of phytoplankton or cyanobacteria that are there and then the toxin amount in the water. And then since historically the Chowan has had the problems with the nutrients, we also will be looking to see if those excess nutrients or if excess nutrients are increasing the amount of toxins that are presenting here. So we'll be looking for associations between those environmental parameters and toxin production. We'll also be looking at a handful of other environmental parameters to see if the relative humidity, the wind speed, the wind direction, the air and water temperature, et cetera, increased or decrease or affect the amount of toxin that we're measuring in the air in general. So in order to sample, I will be collaborating with the NCDEQ water monitoring team. So they're gonna be collecting samples for me with the water. They'll be collecting water samples and also air samples with these small vacuum pumps. So the vacuum pumps just run at a set pace. They pull air across a filter, and then we can extract the toxins and cells from that filter and then quantify the amount of toxin that is present in the air. So those will be deployed by the NCDEQ as well as a group of citizen scientists from the Chowan Edenton Environmental Group. So there's a handful of people living in this area who have been helping out in a couple labs at IMS as well as around the coast. And they're going to be collecting water samples and air samples in their backyard. So I'm really excited to work with these citizen scientists. A lot of them were actually scientists when they were younger, and now this is a problem that they want to get involved with and they want some more answers to. So if anyone who's listening is interested in participating in these types of water quality monitoring projects, there's all sorts of opportunities for citizen scientists who are living on the coast or near these types of bodies of water. And the reason that we use citizen scientists is because it's a great way for scientists to communicate with the public, but it also helps us broaden our reach and the consistency of our data collection. So, of course, these people who are living on the Chowan River, it's very easy for them to just go out to their backyard and place these air sampling pumps. So this will be a great collaboration for us, and the people who are working with us will be certified to collect a whole variety of data. And another part of this project that I am really excited about is the outreach component. So when I was in high school, I was in a special film program that allowed me to become really interested in documentary making. And I think that documentary is one of the best ways that you can reach an audience. So it's so much easier for people to connect with conservation issues if they can see what they're working with and if they can put a face to a scientist. So we will be using an undergraduate intern from UNC Chapel Hill's journalism program, and that intern will be living and working over the summer in Morehead City with me, and we will be creating a series of short videos, kind of like crash course videos, about water quality in North Carolina, cyanobacteria blooms, factors that promote them, et cetera, and then we will be sharing these videos all over social media. And then the intern will also have the opportunity to create a short film or documentary of his or her liking that they can then submit to various film festivals. So I'm really excited for the different little video projects that will come out of this project as well. And then finally, future direction for my PhD in general, since I'm just getting my start in my first year, I hope to create a better understanding of the physical and chemical factors that may promote cyanotoxin aerosolization, especially in North Carolina, because it could vary by region. And I do want to study the differences in those microcystin side chains, as I mentioned earlier, because I think that these side chains could influence the toxin's ability to become aerosolized. So as we talked about with red tide toxin, brevetoxin, the reason that that toxin is so easily aerosolized is because it's very hydrophobic. So when these red tide cells are broken up at the wave break in the ocean, they can easily adsorb to the sides of these air bubbles that are entrapped by the waves. And so that's why the brevetoxin is so easily aerosolized. We know that very well, but we know very little about the different types of chemical factors that may promote the aerosolization of these cyanobacterial toxins. So that's something that I'm interested in pursuing further. And we also want to better understand, are these toxins being aerosolized extracellularly or intracellularly? So those are some other topics that I would like to look at. So I just want to say thank you to all of my lab mates, everyone who has supported me up to this point and helped me get here. And also I want to say thank you to AppNep and NCC Grant for the opportunity to pursue this research under their Estuary and Research Fellowship. And also I want to say thank you to my committee and of course the awesome collaborators at the NCDEQ and CEEG who have helped me create this project and bring it to life. So without further ado, I'll take questions. Thank you for letting me ramble on about algae.
Let's give Haley a round of applause. Thank you very much. All right, yes, let's take questions. If you're down here with us on the first floor, wave at me if you have a question. I'll bring you a microphone. That helps everyone in the room to be able to hear. If you're on the second or third floors, you can ask questions too. Just wave down at us, and then we'll call on you and you can yell at us, and one or both of us will use the microphones to repeat the questions, make sure everyone can hear. So if you've got something on your mind right off at the beginning, let me know. Wave at me. Otherwise, okay, I'll go. That's fine. So I'm thinking about the, yeah, this mic doesn't work well, does it? What we've seen cyanobacterial blooms in this region, a lot of them in the past. You said agricultural runoff. Do we see historical trends in breathing problems, or are people going to ERs?
That's a great question, and that's definitely something that I've thought about. Is this even a problem we need to be looking at? Because no one's complaining about it. But since microcystin specifically acts in the liver when it's traditionally ingested, which is a pathway that we well understand, I think that maybe we're not seeing as much effect from the inhalation of this toxin because it might be acting differently when it's inhaled. So as I demonstrated with that mice study, they were only seeing respiratory irritation when microcystin was inhaled in the mice. They weren't seeing the impact on the liver or the impact anywhere else on the body. So maybe that's why we're not catching it. So that's something that I think could be explaining why maybe we're not getting as many complaints, but it could be having health effects otherwise that we're not catching.
I guess you answered all of their questions. There's one.
Presumably, these toxins evolved for a reason. What are they adaptive for? What do they do?
That's a great question. And if you had the answer to that question that could answer this in every system in the world, then you would be a very rich man. So we don't necessarily know why these toxins are being produced. It is suggested that there is some ecological significance. So as I mentioned briefly, it might be to protect these cyanobacteria from grazers, so small zooplankton that might be consuming them. And then it also has been hypothesized that, say it could be interspecies competition, that they might be outcompeting other types of cyanobacteria that are growing. So there's a couple questions that are being thrown around and a couple theories that have been thrown around. There's definitely an extensive list. There's the other PhD student in my lab, Malcolm Barnard, is actually looking at this and how nutrients could be influencing the amount of toxins that are being produced. So if you're seeing excess nutrients, these cyanobacteria might have extra energy to put into toxin production, and then back to the grazer problem as well. This other PhD student, Malcolm, is also looking at that problem.
Would you expect that if you do see aerosolation with cyanobacteria, do you think that that would hasten management efforts like regulating nutrient runoff or whatever the sort of different source pollutions might be that lead to those blooms? Like, what would be the management implication after that if we know there's an added public health impact?
That's definitely a good observation and question. I didn't mention that we don't have any sort of regulation for the inhalation of microcystin. We have standards that the EPA and the World Health Organization have set for the ingestion of these toxins via contaminated drinking water and foodstuffs, and also just dermal contact. But there are no regulations yet for the inhalation of these toxins, and that's also something that we're hoping to inform with our studies. And as far as would this hasten our response to it, I definitely think that when you take environmental challenges, environmental problems that we're having, and you can tie it back to a way that it's influencing human health and human populations, then that definitely ups the care that most people would have. So I like to say, I guess my loyalty doesn't necessarily lie in protecting human populations. I've always been a huge animal lover. I studied marine science for my undergrad, so I'm really passionate about conservation is really why I'm up here standing in front of you. But I found out that if you convince people that they need to be saved by this conservation issue, then that can really hasten our response to it. Don't tell anyone I said that.
It's on the internet now.
Haley, do the community members have any guesses about why there's this increase in blooms in the Chowan now?
This is actually something I definitely want to interview people about when we start making those short videos in that documentary. I've only been living in North Carolina for about 8 months, and I'm sure that the people with the Chowan Edenton Environmental Group have their theories and they have their observations over time, but there's none that stand out in my mind right now that I've had a conversation with anyone about yet. But stay tuned for the documentary.
Haley, do you plan to correlate the presence and concentrations of the airborne toxin with algal species type and concentrations? And a second question would be, what's known about like the persistence of the aerosolized toxin?
Yeah, okay, great question. So to answer your first question, is there an association between the type of cyanobacteria and its presence in the air? And I think that the morphology of the cyanobacteria definitely could influence its ability to become aerosolized via that bubble bursting. So of course you have certain types of cyanobacteria that are filamentous. They're more stringy. And then you also have cyanobacteria that are just tiny circles. And I think that this definitely would have an influence on, is it on the surface of that bubble that's bursting? That's definitely something that I would like to look at some point throughout my PhD, maybe not necessarily in this specific project, but it's a very good question. And then, I'm sorry, remind me of your second question. Persistence, right. So we know that in the water, microcystin generally has a lifetime from 4 to 14 days. So it doesn't stick around for too long, and I can imagine in the air it might degrade even quicker, and that especially depends on, is it in the cell, is it outside the cell? So that's something that needs to be studied further.
See one on the second floor.
Yes, is there any data that's been discovered about uptake of these toxins into plants on land or animals on land?
Is there any data on the uptake of the toxins in plants or animals on land?
I believe there is some data on the uptake in animals. So this has definitely been studied in various wildlife and also livestock. But I do not know of any data of the uptake into plants.
Let's give Haley one more round of applause.
Thank you all so much.
Thank you for being here.
Yeah, absolutely.
Thanks for sharing. It sounds pretty exciting. You're at the breaking point. You're at the breaking news level on the edge of the science.
I hope we're wrong.
Well, yes, I guess we should.
Public health.
Well, if you're wrong or right, I think we should have you back, and you can give us an update after next summer, perhaps,
Yeah.
on what you found. Hey, everybody. Yeah, I got a thumbs up on that idea. All right. From the Sea Grant folks. That's great. Well, hey, everybody, thanks for being here for this week's Lunchtime Discovery. We'll be back here next Wednesday. Hope to see you again real soon. Bye, everybody.
Thank you.